what I'd like to do today is give you a broad overview of the secretory pathway, what the major steps in the secretory pathway are, and just a general overview of what happens at those steps. So the first step in the secretory pathway is the endoplasmic reticulum, or ER. That's where newly synthesized proteins enter the ER, enter the secretory pathway, and they're then processed and butted off into vesicles for transport to the next step in the secretory pathway, the Golgi apparatus. Now, the Golgi apparatus is generally a stack of membrane cisternae, these kind of pancake-like compartments, where a number of other protein processing events occur. But the main function of the Golgi is to act as a hub for sorting proteins, for routing them to different locations within the cell. It's best to think of the Golgi as like an airport hub where all the proteins, for example, fly into Chicago and people get routed everywhere to places like the lysosome, to different storage compartments, in case of like a number of secreted proteins that you stockpile and then dump into the bloodstream like insulin, or, and most importantly, how proteins get to the surface of the plasma membrane. This is the way that they get routed there. So you have vesicles that are targeted and fused to the plasma membrane, and that's how transmembrane proteins get there. That's also how secreted proteins, all the proteins in your bloodstream, end up there. It's through this exocytosis or secretion mechanism. So that's the general steps and kind of the general overview of how proteins move through the pathway. But what I want to do now is what different functions take place in each of these different membrane compartments of the secretory pathway. So as I mentioned previously, the ER is the gateway to the entire secretory pathway. So that's where new protein synthesis occurs. And proteins are synthesized there, and as a result of that protein synthesis, that means that the proteins also have to begin folding there, and that creates some special problems that I'm going to go into in a minute. So proteins fold in the ER, and then additionally, lipids are also synthesized on the surface of the ER, and that's how lipids are often trafficked through the cell, and that's an active area of research right now, how lipids are trafficked throughout the cell. I'll go into that as well. Calcium is stored in the ER, and that often has a signaling function. And lastly, N-linked glycosylation occurs in the ER. So what is N-linked glycosylation? That is adding sugar groups to asparagine. Now, the N is the single letter code for asparagine. So that is incredibly useful. Knowing the single letter codes for everything is like knowing the alphabet. I use the alphabet every day of my life since learning it. Knowing those single amino acid codes is incredibly useful. So N-linked glycosylation occurs in the ER. That it's mainly useful, at least in terms of the way I teach the course, as a marker for different biochemical processes that were occurring in the ER. Now, as we move on to the Golgi, we also have proteins get processed. As I mentioned, they get sorted. So processing means that they get trimmed and clipped. But the other thing that you'll often see, for example, on MCAT exams or other things, is this O-linked glycosylation. Now, what is O-linked glycosylation? In this case, the O does not refer to the amino acid code. It refers to serine and threonine. And the reason that it's called O-linked is because both of those amino acids have hydroxyl groups. So the sugar is added via the hydroxyl to the amino acid. Now, given these functions, now these are all things that you could memorize, and it's good to know about them, but what's really interesting is what these facts actually mean and the implications they have for how the cell functions. And so what I'd like to do is walk you through some of that now. Now, as I mentioned in my overview, one of the things that happens in the ER is that proteins that are entering the secretory pathway have to fold. And this creates a special problem for secretory proteins because the biochemical environment inside the cell and outside the cell are very different. So the cytoplasm of the cell is reducing, and outside the cell, it's oxidizing because of the oxygen-rich atmosphere. So this has drastic effects on how proteins fold. So you have proteins that normally are in a reducing environment and yet have to be stable in an oxidizing environment. So as a result, the ER acts as a kind of airlock where proteins enter the ER and have to fold in in a biochemical environment that is more oxidizing than what you see normally in the cytoplasm. So that those proteins that are inside the vesicle, when eventually they reach the plasma membrane and the contents are dumped out and the protein is now out in the oxidizing world, those proteins are stable and don't become destabilized by the change in the chemical environment. So what you need is this special biochemical environment. You need a special folding environment, and that's what the ER does. So one way to think about how the ER works is it basically acts as an airlock where you have a reducing environment in the cytoplasm, proteins are translated, moved into the ER, and they fold in an increasingly oxidizing environment so that they are stably folded when they're eventually deposited in the world outside the cell. So you provide a transition system for allowing proteins to fold in an environment that will be conducive to them being stable and wherever they finally end up. Now, another biochemical function that's associated with ER is lipid synthesis, as I mentioned. Now, if all the lipids are synthesized on the surface of the ER, there's a whole set of problems that are associated with that. But one of the more interesting ones and more intractable ones is the fact that there are a number of other membrane compartments that need lipids in order to maintain their integrity. So, for example, mitochondria. So if you're synthesizing lipids on the surface of the ER, how do you go and transfer those lipids in order to support mitochondrial function? And this has long been a problem in biochemistry and cell biology, is how do these lipids get from the ER to the mitochondria? Now, until relatively recently, there's just been a lot of ideas and it hasn't really been very clear. But now there's this very active area of research where it is clear that the ER makes all sorts of different contacts with different organelles within the cell. It reaches out and hugs them. And so one of the ways that this happens is that there are all these ER organelle contact sites. So you'll have ER mitochondrial contact sites, ER plasma membrane contact sites. There's all sorts of different ways that the ER makes direct physical contact with other membrane-bound compartments, and it's believed to transfer lipids directly. And this is something that people are really excited about taking apart. Okay, lastly, my survey of what do all these facts mean, I just want to go back to this issue of protein glycosylation in the ER, because I think this raises a lot of issues of how one part of the course interacts with other parts of the course. So I mentioned that protein glycosylation occurs in the ER. You have N-linked protein glycosylation, but that means that I'm adding sugar groups to proteins in the ER, but somehow I've got to take sugar that exists in the cytoplasm and move it into the ER. So how is that done? That implies that there has to be some sort of transporter system for moving various types of sugars that I use in protein glycosylation into the ER, where then enzymes utilize those substrates and add the sugar groups to various proteins. But that also means, it raises a whole other set of questions. Do I need energy in order to concentrate sugars in this relatively small membrane-bound compartment? Also, as I consume sugar and I throw off byproducts as part of the glycosylation system, how do I get rid of those byproducts? Do I have another set of transporter systems? So we often think of the ER as just being this membrane bag where things happen, but there's a lot of interchange and transport across it. And that when you learn a simple fact like protein glycosylation occurs in the ER, there's a whole set of different systems that we've already learned about in the class, or that we will learn about in the class, that you realize that all these systems have to act in concert in order for just one of these simple functions to take place. So when you learn about small molecule transport, we usually learn about it in the context of ion flows or various other things, but those transporters don't all have to be getting things out from outside the cell into the cell or vice versa. They can be moving things from within a compartment into the cytoplasm, or from the cytoplasm into the compartment.